Okay, hello everyone. We'll get started. Before I want to start, I want to say thank you everyone for coming here. I hope you guys are excited for the workshop. So we'll get started. So I'm Sapo, a PhD student at the University of Oxford. I use computational neuroscience to model the brain using the human connectome. Today, I would be co-leading the workshop too with Arushi Varden. Arushi, would you like to say hi to everyone?
Yeah, sure. Hi, everyone. I'm Arishi. I'm the co-chair and the research lead for this competition, and I work with Fly Connectomics for my master's thesis.
Yes, that's great. And she's also the academic ambassador of biological sciences at Clematis. And as she mentioned, she's a graduate student at the Department of Cells and Systems Biology at the University of Toronto. And as she mentioned, her research focuses on the connectome-driven exploration of neural connectivity in the Drosophila visual system. And in fact, the image that we see here is of the Drosophila visual system. And it's also the dataset that we'll be using for the competition and our workshop. Okay, our workshop will have two parts, a presentation followed by a live demo. So I would be doing the presentation and Arushi would be showing a very cool live demo of the powerful connectomics tools. We hope that after our workshop, you feel more confident that writing research report and exploring the connectome is an achievable goal. And that is important. By the end of this workshop, we hope that you feel like it is possible to actually explore a large-scale dataset on your own. So I will go a bit about what the competition is, and I'm sure some of you may already know, but some of you might be new, so just bear with me. So very recently, neuroscientists, computer scientists, and scientists from around the world have created one of the most comprehensive wiring diagrams of the brain. In other words, the study of how neurons connect to each other is called connectomics. It's called the field of connectomics. And this comprehensive wiring diagram is called the connectome or the brain map. And we will be using this very fascinating data set to uncover new insights about the brain. And together, Arushi and I, and on behalf of our faculty partner, Professor L. Gruntman and Ina Anreiter, want you all to be a part of our learning journey to use powerful connectomic tools to visualize and analyze the connectomics dataset. So in other words, we will actually be using two powerful tools in analyzing, a large-scale brain dataset. The first one is called Neuroglancer. You use Neuroglancer to actually visualize this complex brain data. And think of the brain as like a 3D image, right? But within this 3D image, you have different slices of the brain. And within this brain, you have like 50,000 neurons and so on. So actually, a group of scientists have created a very cool platform to actually visualize neurons and look at its structures. And today, Arushi and I will be showing you how this Neuroglancer is such a cool tool. Next, we'll be also using neuPrint. So neuPrint is a tool for querying neuronal connectivity. But you might be like, why do we need to, like, how is it fascinating to actually query neuronal connectivity? Well, just to keep in mind, the optic lobe, which is the area related to visual processing in the fly, this optic lobe alone has over 53,000 neurons, and they have classified it into 700 plus neuron types. And together they form 49 million synapses. In other words, there are 49 million connections. So as you can see, it's such a massive data set, and covering a small part of this neuronal connectivity is something that connectomics researchers, neuroscientists do every day. It's such a powerful tool to like gain new insights about the brain. And by, studying neuronal connectivity alongside with actually visualizing how the neuron is in a 3D space, you can learn so much about the brain. An analogy I would like to use is like, you diving into a vast ocean to find how thousands of species like, plants and fish interact with each other. And I would be doing the presentation just to give you like a guide, and Arushi would actually be doing a live demo. But what's the challenge for the competition? And I will come back to this again and again, but just to give you like a goal of what we'll be doing. So the first step is we'll be choosing a neuron type. And choosing a neuron type, we can start with actually using the tool, which is the visualization neuroglancer tool. But there's actually different ways to pick a neuron that you like. And again, Arushi will sort of show how to do it. The second step is once you pick a neuron type, you will be exploring neuron morphology. In other words, you will be exploring how the structure of the neuron, you'll be exploring where in the space of the brain does this neuron traverse? Like, does it go from region A to region B? And where does it take input from? Where does it output to? Those are very important in informing how this neuron work together to, induce a behavior or process of information. Step 3 is to, once we visualize the neuron type, we will now analyze the connectivity using neuPrint. In other words, we want to see this particular neuron that you choose, how does it connect to other neurons? From which neuron does it take input from? And to which neurons does it output towards? Okay, so we'll be analyzing connectivity using neuPrint. And lastly, you will combine what you explore using the connectomics tools, and then you will actually go search the literature, and together you will try to propose how your particular neuron process certain visual information or process other information, and in turn, how does that contribute to a behavioral response? So I know some of this might not make sense right now, but we are gonna spend the entire presentation talking about this. In the end, once you do all of the research, you will submit 2 final deliverables. The first is a creative piece. We want to see that you're having fun with this project and that you actually can communicate what you have learned to a public audience. So you can create a creative piece. This could be a video, art, post, infographics, and whatever you like. It can be a poem, it doesn't matter. But the idea is you create a creative piece. The second part is you write a written research report. So it'll be like a 500 to 1,000 words written research report on a specific neuron and how you use like the connectomics tools to gain new insights about how neurons contribute towards inducing a fly behavior or processing certain information. Okay, all of these are in our guide, but I want to point attention to this particular thing, which is the early feedback opportunity. So our final submission deadline is December 1st of 2025, but for everyone who, try and actually do the connectomics research and try to write a report and do the creative piece, if you guys do it before, if you submit before August 31st, we would actually give you feedback and so that you can improve upon it and submit a better version by the final deadline. And we will announce the winners January 2026. You might be wondering, how difficult is the field of connectomics and computational neuroscience? Is it really easy for anyone, regardless of their knowledge background? The answer is that yes, it is. We think it is achievable and that it's very possible to explore such large-scale data set. However, just, generally for research or science, it does not mean that that's something you can do in 5 minutes, right? Like everything, it takes time, but the point is it's achievable and that it is possible to do it. However, we also recognize that everyone comes from different backgrounds and we have learned a lot from you guys, invaluable feedback from workshop 1 and the types of questions that were asked during and after the workshop. So taking all of your feedback, we have curated some learning guide and resource compilation. The idea behind this learning guide and resource compilation is very simple. The idea is that we want you to have a clear learning goal so that you know how to get there. But what do I mean by getting there? Getting there means you are able to conduct connectomics research, you're able to write a meaningful research report, and you're also having fun with it, okay? And then we'll be using this learning guide or learning objective as sort of like the pillars for today's presentation. And hopefully by the end of this workshop, or hopefully by the end of the competition, you have a clear roadmap of what concept to learn or what concept you would have learned, how to use the tools to explore this large-scale brain dataset, and also believe that you can actually do this. Before I go into my presentation, I want to point you to what I meant by the resource guide. So we have this competition materials. We have 4 files. Arushi, if you could send in the chat the tinyurl.com/flycom2025, it will take you to this competition materials. But essentially we have 4 files. If you click on this first file, you would see it's the competition handbook. It has every single thing from, what the competition is about, some cool images, eligibility criteria, awards, how to participate, what are the 2-part submission or deliverable, the date, selection criteria, and important links, and so on. The second file, which is this one, a starter guide. This starter guide contains sort of like the basic concepts in neuroscience and in the fly brain that you kind of need to know. And then we also have a step-by-step user guide for the Neuroglancer, which is the visualization tools. It's a step-by-step guide. And then we also have the step-by-step guide for neuPrint. Then the third is the research report template. So we want to sort of guide you on how to write the formal research report. Again, you don't have to follow it. These are just the questions for you to think about. The fourth one that we have recently uploaded based on your guys' feedback is this learning guide and resource compilation. And the idea is that we want you to think of this as a learning roadmap. What do I need to know in order to do this competition? what exactly is the connectome? How do I think like a neuroscientist when I look at the circuit? And so in other words, we have broken this into key conceptual building blocks, and then we pair these building blocks with some learning goals and some resources in which you can learn these things. And the idea is that you can sort of check these learning box, and hopefully if you know all of these learning objectives, you are ready to sort of do well in the competition, right? So an example is, okay, of course, to understand the connectome, you need to know the building blocks of nervous systems, and then I have some terms that you sort of need to know, like neurotransmitter, excitatory, inhibitory, and so on. And then we have matching resources, like where can we look for these information? So we have five main building blocks, such as, okay, you need to understand what the connectome is. You need to know at least a bit about the fly and the optic lobe. You need to know how to use Neuroglancer and neuPrint, but specifically, what do you need to know in Neuroglancer and neuPrint? We have sort of outlined into concise bullet points. And lastly, how do we actually combine all of this together to produce a meaningful report and to think like a neuroscientist? So, anyways, without further ado, I have made that learning guide into a presentation. So hopefully after this workshop, you go back and look at the fourth file, which is the learning guide, you sort of understand much better what's going on. Okay, so let's get started. So as we see here, we have five learning objectives, and we'll go one by one. The first to understand is we need to know what are the building blocks of nervous systems. And of course, there are many terms, but I want you to be familiar, especially with some of these terms. So this is a neuron Arshi and I have screenshotted from the Neuroglancer, which is the visualization tool that we'll be using. I have screenshot this neuron, and you can see there's a few terms that you sort of need to know. So first, a neuron is sort of a cell, a cell, right? And what type of neuron this is? If you go in Neuroglancer, you would see, this is a C2 type neuron. So the name of this neuron is C2. The second step is to know what chemical messenger does this neuron use to communicate with other neurons. So actually, if you go to Neuroglancer, which again, Arushi will show you in detail, but you will see it's a GABA neurotransmitter. And then if you go to the learning resource guide, you would know GABA, I believe, is inhibitory. And you're like, okay, this neuron performs an inhibitory function. Another key terms you need to know is pre-syn and post-syn. When you go in Neuroglancer, you can actually see where the neurons receive inputs. So that's labeled in blue circles, which is in this neuron, does the neuron receive its inputs from other neurons? So that's in the blue circles. And then the pre-syn means that this particular neuron is at this particular synapse, is sending output to other neurons. So that's what this means. And again, Arushi will sort of explain again and again and again, but this is just sort of the learning objective so that you can get familiar. So once you understand the building blocks of the nervous system, the second step is to understand how do you actually reconstruct synapse level connectome. We don't expect you to know every algorithm, every detail, but I think it's good to sort of appreciate what goes behind in building such a large scale data set that you'll be analyzing. And keep in mind, we started with C. elegans, which is a 300 neurons worm. It's a worm, right? That also took quite long. But then 20 years later, only in 2024, last year, there have been this revolution in neuroscience, where they have been able to create one of the most comprehensive wiring diagram of the fly brain. And the optic lobe that we can analyze has over 49 million synapses. So can you imagine how fascinating, how large scale that is? But how did that happen? First, you actually take a slice, like literally you slice the brain and then you take the microscope and then you get these images. Okay, it doesn't mean anything much, but you can see each of these space, like the red dot, is actually a neuron. Then what you do is you take, let's say, I don't know, thousands or millions of slices, and then you sort of put them together, like as you can see on the image at you can see here at the image here, you see, you take many slices, and then you sort of get like thousands, 10,000, millions of images of these blob looking like images. So these are called EM images. Okay, then they use some sort of algorithm to solve it, like, okay, this red dot is actually belongs to this neuron. And then actually there's a cool visualization video in our resource guide. I would encourage you to have an animated look at that. It's so cool. Like within each slice, you can actually have an algorithm that traces the neuron slowly. And then you're like, this is where the neuron is in space. So anyways, the goal here is to not fully explain how they reconstruct the connectome, but just to let you appreciate like what's going on. And of course, once you do this for like 10,000 neurons or whatever, they created a tool like Neuroglancer and neuPrint to actually analyze how neurons look in the 3D space and how it connects with each other. So, which is what we're going to analyze. That's what we're going to do. The third learning objective is to understand the optic lobe. Okay? At the very minimum, you need to understand as soon as the fly brain, or as soon as the fly, it has big eyes, as soon as the fly receive light input, like a visual scene, how does this visual information travel through the brain? Or in other words, what are the brain regions of this optic lobe that help process this information and lead to a behavioral response? We call these neural pills, okay? And there are four main ones. So the eye is not shown here, but once assuming the light passes through the eye, the retina, the photoreceptors, and whatever, it passes through four main regions. The first is called lamina. It's written incomplete because, Arushi, you can correct me if I'm wrong, but the data set that we have, it perhaps does not have every single neuron in the lamina, right?
Yeah, this is not the full construction of the lamina. It's just some parts. Yeah.
Exactly. But it shouldn't matter for our purposes. But just know, like, there are four main regions, lamina, and then medulla, and then lobula and lobular plate. So throughout the combination, make sure you memorize the name of the neural pills and if possible, try to memorize the shape as well, because we have taken screenshot from the Neuroglancer, which is the visualization tool you'll be using. But again, all of this, it's in the starter guide, you see? Lamina, medulla, lobular plate, and then we have like a lot of details. And we have also covered this in detail in the workshop one, which is on our YouTube. And Archie, can you send a YouTube link to our workshop one? Thank you. Cool. Once you understand the optic lobe, the next goal is to be able to use the Neuroglancer and the neuPrint. But let's focus on the Neuroglancer. And keep in mind, Archie will cover this live, but I'm showing you sort of like what you need to know. Okay? So the goal of using Neuroglancer is to visualize the neuron and explore the neuron morphology. So when you go to the actual Neuroglancer, you will see different terms. And so just to get you familiar with the terms, I've labeled some of this here. Like I said, there's, I don't know, 50,000 neurons or more in this optic lobe. And each of the neuron is assigned an ID. Okay, so 71045 is the ID. Perhaps, so that's an individual neuron. But then we also have something called label. So these labels are labeling neuron types. Okay? And so, like, imagine if you go to Walmart or like a supermarket, you see, let's say, 1,000 packets of chips. But within these 1,000 packets of chips, you will see different types of chips, like Lay's, or you see, I don't know, Cheetos. So Lay's, Cheetos, these are called labels. They are types of chips, right? But a same neurotype may actually have different neuron IDs because there could be, I don't know, 10 C2s or 100 C2s, I don't know. But each neuron type can have many, many different neurons. Okay, for each of the neuron obviously have to communicate with other neurons. Remember from the building blocks of nervous system learning objective? So therefore, in Neuroglancer, you can actually see, okay, it seems like C2 actually uses GABA, which is an inhibitory neurotransmitter. So it would be here, NT stands for neurotransmitter. Okay. Then as I've shown you before in the building blocks of nervous system, so ignore this layer for now, but you can see that if you click on some of the thing, you can see where the neurons is receiving inputs and projecting its output towards, like where in the neuron. So this is marked by a red and I guess bluish circle. So a part of using Neuroglancer is to explore where in the neuron does it receive inputs and send outputs towards, and there's a way to do a Neuroglancer. And then, importantly, you also should know how to understand the layers. So remember, we have like a neural pill. So you have four regions. For each of the neural pills or each of the regions, we have different layers. It would be cool to see that, okay, this particular neuron receives, let's say, input and output around layer 5 of the medulla. So medulla is the neural pill, is the four main regions. Layer 5 is just one of the 10 layers that medulla have. And in the report, we would like you to understand how neurons traverse through the brain regions and use neuron terminology such as layers and neural pills, okay? And there's actually a way to look in the Neuroglancer, and I've screenshotted here, but then when you actually go and explore yourself, and Arish is going to show as well, it will be sort of clear, hopefully. So that's sort of what you need to know in the Neuroglancer. In the neuPrint, so remember Neuroglancer is a visualization tool. NeuPrint is to analyze neuronal connectivity. So let's take a random C2 neuron. So the ID here is 100043. Okay, that's the ID, that's the neuron times. So now we want to see how many input synapses does it receive. So it says 117, and then it apparently has 54 synapses that it sends output to other neurons. Okay, so these are the numbers. And then you can also see like the brain region breakdown. In other words, it receives input. So this neuron receives input at 99% at this medulla, the right medulla, and so on. So, and Archie will go into detail. And again, in the neuPrint, you can actually again see the neurotransmitter, and like the Neuroglancer, it shows its GABA. When you actually click this C thing here, so this C, you can actually see sort of like a pie chart. So I'm gonna zoom in, but essentially this graph shows like upstream. So upstream means from which part of the medulla does it receive, or which neuron does it receive inputs. So here you can see, okay, it receives from L5, from L1, and all of these are neuron types, by the way. If you notice here, all of the neurotypes are actually in the medulla. But the question is, where does this neuron downstream its output to? So again, it says, okay, it seems to be outputting the most to L1, L5, MI1, L2, L4, TM1, and so on. You can explore this for all of the neurons, and it's so cool because it's such a large data set, you can explore whichever neurons you want. Now you have understood the optic lobe, the basics of neuroscience, and how to use the tools. Hopefully, at least you get the main gist of it. But how do you actually put all of this together? And that's the question we sort of sense that you guys have asked from the workshop one, and it's completely understandable. So to make things very accessible, we have tried to break down into three main components of this neuroscience research, or at least for the competition. The first part is, imagine when a fly is looking at a visual scene, or it's like I'm looking at you right now, like on the zoo. So whenever I look at something, my eyes or my brain will capture some features, okay, some visual features like size, shape, and so on. The second step is, once you see these features, my brain will want to respond in a certain way based on whatever I'm seeing, right? It sort of makes sense. If I see a truck coming, like a car coming, I would run away. That's the behavioral response. And the third important thing to realize about this competition is that through all of this processing information and behavioral response, there's a neural mechanism behind this, and there are different sets of neurons that work together. And using the connectome, we can really uncover to the T, like how is this neuron working together to produce some of these features and behavioral response, okay? Or at least we can use the computational data to predict things. So what do I mean by identifying visual features? So imagine this is a fly, and you see this fly has such a big eye, and that's why it allocates most of the stuff to visual. And then let's say there's an object flying, but it's now flying down. Can anyone tell me in the chat that when there's an approaching object and the fly, so let's say we have a fly that's stationary, and then you have an object that's approaching, can you identify one visual feature that changes in this process for the fly brain? So I'll repeat, what are the visual features that is associated with the object approaching? Okay, another animation. What does that mean? 222. No worries. Background, sure. But think of like, you're getting close. Yeah, background. But what about the bird itself? Brightness, okay, brightness is one. Awesome. So you're correct, brightness is one of the visual features. But what is the very obvious one? How do I know that the truck is approaching me? Size, awesome. So actually size, brightness of course, but the size is the main feature that happens when an object approaches, right? So these are visual features. So when you read a paper, you will see these different visual features like size, shape, brightness, color, position, velocity. And a change in some of these or a combination of these features give rise to a complex visual phenomena. So for instance, an approaching object would change a variety of features such as maybe brightness, velocity, size, shape, or whatever, right? And together, we combine all of these change in features and we call it looming. So this process is called looming, and it's the way of describing how something becomes bigger, like to me. So that's looming. So I hope you understood the first point is we identify visual features, and you will see this in so many connectomic papers, at least for the fly. The second part is once a fly or once even a human being, right? When we see something, we will maybe not all the time, but we will often have a behavioral response to it. And especially flies, like imagine I'm trying to hit the flies on my hand, it escapes right away. Like, how does that work? So that is essentially a behavioral response. So the first behavioral response that when an object approaches to the fly is that it flies away, it takes off. So an example is it flies away. So that's the animation. Or the second is maybe it's flying and then it tries to land. So that's another phenomena. And let's say something is slowly approaching, maybe the fly would walk backwards. Apparently that's a thing. And then you see the fly slowly walk backwards. And as you can see, that's as important as it is to detect some visual features, the fly needs to actually do something. It needs to take an action. Otherwise, it won't survive, right? If it sees an object that's approaching and if it doesn't actually do the behavioral response like taking off, the fly will not survive. So the features processing information, it's usually coupled with a behavioral response, and that ensures that we as a species survive, right? That's just the nature of animals. And but the third thing is that from processing information to actually inducing a behavioral response, there's a neuromechanisms, and that different neurons work together. But of course, we know neurons work together in every sense to give rise to our behavior and so on, and cognitive processes. But when you go look at connectomics papers, you will see different depictions or visualization of these circuits. I want to show you two today. So hopefully once you see these figures, which is, I think it will be quite often that you see these figures, when you actually go and read them, it will be more familiar. Let's see this paper. So can anyone tell me what neuron type is this pink neuron here? It's in the diagram. So can you name this pink neuron type? The information is in the figure. Great, it's LC4. And you know, these neuron types are actually, you can visualize it using Neuroglancer. So you can see, can anyone guess what does ME stand for? Remember, we have talked about 4 regions in the optic lobe that is there for the competition. Can anyone guess what ME is? Great, medulla. Awesome. So just to recap, we have 4 neuropils. Lamina, which is not shown because I guess it's not relevant for this particular circuit, but we have lamina, which is on the top, and then we have medulla, and then you have lobula, which is LO, and we have lobular plate, which is LOP, and all of these acronyms and stuff, it's all there in the guide, but this is just to like get you more familiar with the thing. Okay, so how do I actually interpret this? Oh, and actually, let me describe OG. So OG is optic glomeruli, and this is not a part of the optic lobe of our dataset. So, you know, like sometimes a neuron in a specific optic lobe can actually project to other brain region, such as the optic glomeruli. So you don't need to care too much about the other regions, just focus on the optic lobe, but just letting you know some of the neurons project to different parts of the brain as well. But for our concern, for our purposes, we'll focus on the optic lobe. Let's see, let's say LC4. So how do we do this? So we can see that LC4, it has some dendritic arbor, which is sort of the areas in which it either receives input or send outputs from. So it forms some input or output synapses from the lobula, and it seems to also be connecting to the optic glomeruli, okay? And it also seems to be very close proximity to LPLC2, which is again another neuron type, which can also be found, I guess, in the optic lobe as well. Okay, so again, you can say the same thing for LC16. It's somehow coming from lobula and going to optic glomeruli. And then you have this T4, T5. T4, T5 are different neuron types. However, maybe it's very similar and in close proximity, so they're just combining it, but actually it's a separate neuron types. But as you can see, this T4, T5, it touches the medulla, it touches the lobular plate, it also touches the lobula. So it seems to be all over the place. So this is sort of what you can see in this diagram. However, we can't actually, unless you know the literature, you can't clearly tell the direction of the connection. Like, does it actually receive input or does it send output? So that is something either you know the literature, know how the eye works, or you actually use neuPrint to analyze the neuronal connectivity. But anyway, so that's one type of diagram representation. Another very common way of depicting neural circuits is something like this. So this is exact same thing, but now it's done in a more like presentable like chunks. So remember as we shown, okay, it seems like T4 neuron originates from medulla and then it projects to lobular plate. But where does it connect to? Oh, it seems to be connecting. Yeah, it seems to be outputting, T4 seems to be outputting its projection to LPLC2. Okay? And what does LPLC2 do? LPLC2 seems to be taking inputs from the lobular plate, and specifically the T4 neurons and T5 neurons. The LPLC2 also seems to be taking inputs from some unspecified neurons within the lobula, as you can see here. And remember, you can always explore and verify all of this in neuPrint and Neuroglancer, and that is why these tools are so fascinating to use. We can also see LPLC2 actually projects to OG, optic glomeruli, but we wouldn't care about this because we are currently looking at the optic lobe. Okay, but essentially the point is that all of these neurons play a role within a larger circuit to do something. So for instance, it seems like LC16, which originates from lobula, plays a role in backward walking. LC4 maybe does something to do with detecting velocity, and LPLC2 does something to do with size, and T4, T5 probably also plays a part of that circuit. Okay? So again, this is just another way of seeing the diagrams. And when you read the literature, you will see many of these, and it's sort of your goal to look at three things. Look at the visual features, look at the behavior, and look at how a specific neuron in context of a larger circuit help all of these processes. So now that we have this all together in one place, let's come back to the challenge. So the challenge is first, we will choose a neuron type. And again, Arishi will show how you can do this in different ways. The second is to describe the neuron morphology. In other words, where is it in the brain? Is it in the lobular plate, the medulla, and so on? And what does it actually look like? So like the stuff I showed you in one of the learning objectives. Then of course, the third is to explore how your neuron actually connects to other neurons. You can use neuPrint to do this. And lastly, we will now bring it together to propose how your neuron contributes within the context of a larger neural circuit to processing certain information and also to producing behavioral response. And in the end, all of this will culminate into a creative piece, which you'll submit through Discord, and then a written research report, which you'll be submit through a Google form. And all of this link is in the competition handbook. Great. And now Arushi will take it on from here.
Okay, thank you, Safa, for guiding us through what the competition's about and the different ways we can um, play around with these neurons and things. I will be doing a live demo on how this research journey might look for you. So let me just share my screen, and I hope everyone can see it. Let me know if you can. Yeah, that's good. Okay, I'm gonna zoom, it's fine. Okay. So firstly, if you want to play along while I'm doing it, I suggest having a few things open. So one of the things I'd have open is this application competition materials page and going into the learning resource guide. And if you go down, another thing I would have open is this connectome paper. This is the paper that's based on the connectome we're using, which is the optic lobe connectome. So that's open here. And if you go down, there's a supplementary file page, and this will be quite useful. And this is something I use a lot in my own research. So it looks something like this. I personally have it downloaded. I like using it this way, so I'll be using that. But this is how you access that. And there's this review that actually supposed to be referencing a lot throughout this talk. It is the photons to behaviors, neural implementations of visual behavior in Drosophila. I would have this review open. It looks like this to help guide you. And obviously having Neuroglancer and neuPrint open. So now that we have all our toolkit out here, let me demo a scenario where I'm a student and I don't really know where to start. Like, I don't know what neuron to choose. It's kind of all getting over my head. Very fair. One of the ways I would suggest doing is going to this review. It's a really nice review that sort of catalogs the main vision-mediated behaviors in the fly. And I would suggest actually like reading it slowly and going through each figure, but there's this big figure at the end that sort of has all the major neurons involved in vision-mediated behaviors. So like something like this. So you see the retina, lamina, medulla, lobular blade, and everything below this. This is all in the brain of the Drosophila. Our optic lobe dataset doesn't cover that. So the neurons we will be interested in will be everything like in this segment over here. So looking at this, if I'm just, I guess shopping for a neuron of interest, let's say like LPLC2 interested you from suppose presentation on this looming thing. It was pretty cool to you. So I want to investigate LPLC2. So then what I would do is I would go into Neuroglancer and okay, just reiteration of the previous workshop that I gave. You will click on the eye to get the ROIs. So these are the different neurophils. Maybe it'll be nice to have a bit of a quiz time. Can anyone tell me what this purple region is in the chat? Take a guess. And the reason I'm asking this in such weird confirmations, close, is because it's good to have an intuitive sense of where these are because there are no labels on Neuroglancer. So one hint, okay, yes, lobular plate. Now, for those of you who are like, how's that the lobular plate? That looks like something else. The lobular plate, it's one way to orient yourself on Neuroglancer is like this. The lamina looks like a molar, like your tooth. It's like chopped off, it's incomplete. So you can like situate yourself that, okay, this is the lamina here. Then one step below the lamina is the medulla. So if you go to back to the resource plate and go to the starter guide, I have labeled these for you here. So this is one place. But unfortunately on Neuroglancer, you actually can't label these. So this is the way I'm teaching is one way to orient yourself around this. So this tooth looking thing is the lamina. Fine. Below the lamina is something called the medulla. And in my head, I find the medulla the biggest part of the eye. Like it's if you see relative to everything else, it's this massive oval chunk that's over here. Okay, so we've got two. Then this is the lobular complex. So how do I differentiate the lobule and the lobular plate? The lobular plate has a clue in it. It looks like a plate. And if you look at it, it looks kind of, I think in my head, it looks a bit like a plate. So it's a lobular plate, and this is the lobule. So this is one trick I have of orienting myself since Neuroglancer doesn't have labels, and it might help you when you're exploring a neuron. So I hope that helps. Anyways, so now that I have all the ROIs ready, my neuron of interest right now was LPLC2. When you're writing the query here, make sure your things are capitalized and not capitalized appropriately because it is sensitive to that. And let's click on one of the LPLC2 over here. If you click all of these, it's going to show you everything. You just want to see one. Okay, now let's reorient yourself. Now I'm investigating this neuron. Okay, it looks like it's going in a lot of places. And how do I know that? So one of the things you would look for when you're trying to investigate this neuron's morphology is look at its dendritic arbors. When I say dendritic arbors, what that means is these like spider web looking things here. Dendritic arbors are where this neuron gets input from other neurons and sends outputs to other neurons. And if it's a bit hard for you to see, like I can't really tell, like it's a bit hard for me to tell where the dendritic arbors are, you can click on this pre-syn option over here. The pre-syn option, as Sapo has mentioned in red here, is all the connections the neurons is sending out. And if you click on the post-syn option, you can see all the connections the neuron is receiving. So now if I look here, I can see, oh, this is where like most of the dendritic arbors are localized here in the lobular plate and the lobula. So that's one part of our investigation we've done. We've found out this neuron transverses two neuropils, which is the lobular plate. Note, I had to kind of memorize the neuropils because there's no label. So now I know, okay, this plate-looking thing is the lobular plate, this is the lobula. And that's because most of its dendritic arbors are localized in both of those places. And it transverses those areas. Okay, now let's go into the layers intersecting. These are two big chunks of the neuropil. Where exactly in the neuropil is this really intersecting? It doesn't really look like from my eyes. The arbors are spanning the entire lobula. It looks like it's spanning the entire lobula, but let's find a better way to really quantify that. So what I would click on is like, I've now established that this is the lobula. So I would go into lobula layers here, click on the eye and click here. And these are all the layers of the lobula. So I will click on all of them and they'll show up. It might take some time. And this is a bit of like, just playing around and figuring out what's covering, what's not. So slightly, I'll peel those layers off. Let's take layer one off. Okay, it seems like that one wasn't really covering anything, so that those arbors are not really in that region of the lobula. Let's take layer two out. I don't really see any of those arbors, like these sort of things showing. So, okay, maybe let's peel another one off. Even here, like it seems like, yeah, there's nothing. So it seems like this neuron is more towards the end of the lobula, not the first few layers. Now let's peel layer four out. Now we're seeing some of those arbors are showing. So that means some of them did go to layer four. So I'll put layer four back on. So now let's go here to the end. So layer seven is the last layer and take it off. So if I take off layer seven, it seems like again it's pretty covered. I mean, this is the tail to the cell body, but there are no like arbors here particularly. And if I take layer six of you see now I've exposed some of those dendritic arbors. So from here I can make the conclusion LPLC2, the dendritic arbors that are localized in the lobula, so the spider webs that are in the lobula, they seem to be transversing layers 4, 5, and 6 of the lobula in particular. So you have deduced that part. Now, similarly, you would investigate the layers that the lobular plate region of this LPLC2, what layers is it transversing in that? So let's go to lobular plate. Lobular plate is designated as LOP. And let's go to LOP layers, not columns, remember, layers. We're not using columns for this competition. So there are 4 layers in the lobular plate. Same thing here. It's covering the entire lobular plate. Oh, I see there's something in the chat. Okay, so we see there are 4 layers in the lobular plate. And again, one by one, we peel the layers and we investigate. And we see, okay, I'll ask the chat now. Do you think I should, this LPLC2 that's in the lobular plate region, do you think it is transversing layer 1 or not? What do you think by looking here? Okay, nice. Well done. Yeah, you can see some of the spidery things are showing up, the places where it's making those pre and postsynaptic connections. So therefore, yes, indeed, it is transversing the first layer. Now let's look at the last layer too. What about this one? What do people think? Is it transversing layer 4 by looking at this? Okay, well done, well done. Yeah, it seems like you guys are getting it. So yeah, so now I have morphologically captured what's going on with my LPLC2 neuron. It has, you know, 2 of its main dendritic arbors localized in the lobula and the lobular plate. And if you want, you can also notice one more thing, like this is the cell body, this little blob I covered in my last workshop, and see where it is going. So it's going right over here next to the brain of the fly. This is the brain, this is the eye. So maybe you could even say, okay, it seems like the cell body is near the fly's brain and something like that. And if you're interested, you can even look at the distribution of pre and post synapses. So remember in high school or like in university, you've learned that your neurons are polar, like there's a really clear presynaptic and postsynaptic site that neurons receive, but that is not really the case in flies and vertebrates in general. And a lot of times you will see a situation like this where those sites are completely intermingled, and it's frankly quite a mess. So if I take out my postsynapse here, you can see that I can't really say like, oh, okay, this is like the presynapses are here, the postsynapses are here. It's kind of everywhere. And if I put the postsynapses back, it's kind of everywhere. So another way to characterize this neuron is like LPLC2's pre and postsynaptic sites are sort of, randomly distributed throughout the neuron or something like that. So that's another way to characterize this. So I have shown you how to visualize this in Neuroglancer. There's one tidbit that I need to touch on, which is the presyn and postsyn. These terms, if you forget, is recent like input, output, or whatever. This is why another reason the supplementary information is very helpful. If you go to the second page or any pages of the supplementary information like these. So anyways, for this, the first one, it says presynapses are outgoing. Means the presynapse is the zone where that neuron sends outgoing connections. And postsynapses are the synapses where it receives incoming connections. So if you ever go to Neuroglancer and you're confused what these stand for, I suggest going to the supplementary information of this connectome-driven neural inventory paper, which is the connectome paper, to have a look at this so you can, reorient yourself to what those words mean. Okay, so now that we've done this, I have investigated what my neuron looks like. Now we head over to neuPrint. Make sure your dataset is Optic Lobe 1.1. There are many datasets, so make sure you're on the right one. Then we go to the search bar and we write LPLCA. And we submit it. Okay, now that I've found myself here you can see there's also a consensus transmitter and neurotransmitter. All of this, again, I touched upon in the workshop. And here you can see the brain regions. So this is the input. So it says the inputs this neuron is receiving, they're mostly localized in these brain regions, the LO, LOP, and there are some places you won't be able to see since they're small. The only thing you have to do is you just have to hover through them. So you see that says PVLPR. So if you're like wondering again, I forgot what LOP means, what LO means, you can just go to the brain regions here and they have explained what those um and yeah, you can see it. So now like maybe PVLP, you're like, I don't know what the hell PVLP is. Like, I've never heard of that. So, and you would be right to be confused. It's somewhere in the central brain over here, and that's something you don't need to worry about. But it's good to know, okay? So your neuron, it seems like it's directly outputting to the PVLP. Anyhow, um, so inputs and outputs. So again, inputs are the inputs LPLC2 receives, outputs are the outputs it sends. Now I want to see what are the top inputs and outputs it's receiving. So in the previous workshop, I told you guys you could go on this C icon that says synaptic connectivity and click on it. And whenever it loads, yeah, you can see where its inputs and outputs are. But if you, for instance, want a really detailed breakdown, you can just also click on this number itself. Also, something, sorry to note, is that there are a lot of IDs and you would be right to wonder, I don't know which one to choose. You can choose any. Remember, there's no, a lot of neurons in the optic lobe, and which is why it's such a great tool to use in science as well, is that there are many repetitions of the same neuron. So you can just click on one and pick the first one and just look at its outputs. So, okay, I'm seeing a bunch of outputs over here and stuff. You can also collapse by type. So instead of seeing a bunch of the repeated outputs, it collapses them by the type of neuron they're connected to. So like you can see these are the different neuron types they're connected to. So that's another way. I personally find just looking at this C synaptic connectivity good enough. It captures the main inputs and outputs. So there will be many ways this diagram looks like. And this is the way I would go about interpreting it. Let's say this is LPLC2. It seems the main input it's receiving. And how do I determine main is by literally the size of this. It corresponds to how big of a percentage of that input it is getting. So it seems like out of all the inputs it's getting, T4A, T5A, T4C, TM5Y, TM20 seem to be some of its major inputs. And its major outputs seem to be LPLC4, LC4, LPLC4 again here. And these are some ways you can understand how your neuron is connected to. Then going from here, I could, now that I know the top input output connections, I could go on Google and I can literally go and type LPLC2 Drosophila. That is a keyword to write because this is a fly neuron. And the first paper I am seeing has something to do with LPLC2, so I'm going to go click on it. So this is the abstract of the paper. So if I go read it, I'm not sure like what's going on with this. Okay. Such as the visual looming motion by imminent collision or approaching of predator. Okay, this is something I'm learning from here. Here we report the discovery of an ultra-selective looming detected neuron. Seems like this paper discovered this neuron in Drosophila and shows some mechanism it does. I'm seeing some other neurons that look familiar, T4, T5. So if I go back to neuPrint, go back to the input outputs, I saw something to do with T4, T5. I saw T4, T5, like subclasses of T4, T5 seem to be a pretty big input to this neuron. So now I'm paying more attention. This seems pretty relevant. And okay, they did some experimental work strongly to something to do with outward motion to the center of the receptor field. Some of these words are confusing, but this paper looks kind of relevant, and you keep reading, keep seeing. Why? Oh, okay. So this covers a wide space, terminates into some descending neurons, and drives the jump muscle to trigger an escape takeoff. So flies use this to discern and escape looming threats. You see, just from as someone who has no idea what this is doing and just sort of reading through it, I've noticed, okay, this paper seems to have discovered this neuron called LPLC2. It has something to do with something called looming. I'm not sure what that means, but I'll go research it. And this is a neuron that flies use to escape some threats or something like that. And this is interesting because I found something to do with T4 and T5 in this. So you see, the abstract already told me so much about what this neuron is doing. So next, what I would do is, okay, I have characterized LPLC2, and I've read this paper, maybe what it does. Now I want to put this neuron in a bigger context. Remember, no neuron works alone. It is in this really elaborated, sophisticated interweb of connections, right? So looking at its input and output, I would like to put this neuron into context. So maybe if you look at LPLC2's literature, you might find it does this in a certain way. Then you go read T4, T5 or LPLC4, LC4 and see what they do. Then you map them all together. You're like, okay, since T4 and T5 do this, LPLC2 does that, LPLC4, LC4 that are downstream does that. I think this is the network it's a part of, and I think this is how it's contributing to that network, if you see what I mean. Something like that. And what you're essentially doing is actually hypothesis generation, right? So for instance, LPLC2 is very well studied, but there are going to be neurons that you come across, and I'll give you some examples. And this is what scientists actually do with this connectomic data set. They look at neurons that we never knew anything about, and they like look at its connectivity and characterize things, analyze things, and then they predict, okay, maybe I think this is doing this. And then later you do experimental work to validate that. So you're at the hypothesis generation stage. And another thing to note is, I believe only 20% of the visual system in the flies neurons have actually been experimentally verified. That is a very low number. And it makes sense because it's really hard to actually do those electrophysiology recordings in the fly. So you will probably come across neurons where, and I will show you an example where nothing is known about. And you shouldn't worry about that. In fact, you should go and take this as an opportunity to try and predict what does this do in the context. And I bet the neuron you choose, probably upstream, downstream, is connected to a neuron that's pretty well studied. Then you can use it to connect. Okay, since that neuron we know does that, and it's connected upstream or downstream to that, maybe this is doing this. And because it's sending output to this, maybe it's doing that. So this is kind of how this research journey would look like. And as I mentioned, this optic lobe connectome came out in 2025. For instance, this paper is in 2020. As I mentioned, LPLC2 is pretty well studied. There will be a lot of papers that are like really old that you find, you can use them. But remember, because the connectome is a new resource we have, a lot of things, input and output connections you see on neuPrint, you might not necessarily find documented in the literature because back then people just had no way of knowing those things, right? You didn't have this resource. So that's something to also keep in mind when you're doing your research. So now that I've shown you LPLC2 as an example, let me give you an example of a neuron that we don't really know anything about. So let's go. So let's say you want to pick a neuron like that's random and whatever, you just want to explore something new. One good way is to actually go to the supplementary information, as I mentioned. And this is a really nice catalog of all the cell types, neuron types, the authors of these papers have defined for us. And I use this all the time as well in my own research. So the way to read this is the cell type or the neuron type. R is to do with right. A lot of the optic lobe is just constructing the right of the eye, and is equal to 1 is how many of these cell types are seen. For instance, AM1, there's only one in the right side, and this is the predicted neurotransmitter. So that is of relevance that you would need to know. So let's say I'm just, neuron shopping once again, and I look, and TMY19A looks very cool. It just looks cool, so you want to find out. Seems like you haven't heard anything about it. Okay, so I picked TMY19A. Again, keep note of how the caps and everything is made. So I go back to Neuroglancer, TMY19A, and I click on one. And then I orient. I won't go over this step again because I went into detail for the previous neuron, but you would see the post-presynapse, you would see the layers it's intersecting, the neuropils it's intersecting to get its morphology. And then you would go on to neuPrint to search TMY19A. Okay, and click on the connectivity toggle and see, okay, it seems like it's getting a pretty main input from T2, and it's sending a bunch of heterogeneous outputs to a lot of places. And I mean, it's getting a lot of additional inputs as well. Okay, so again, I'm just going through this really fast because we already know this process. I've been through it. So now let's search TMY19A neuron. Oh, Drosophila. Seems like, as I mentioned, there will be a lot of neurons that we just don't know anything about. So even if you click these papers, you will actually find TMY19 doesn't actually have anything in the literature for it. So what do you do in this case? You could go and find other ways to explore this. So how I would go about this is I would get rid of the 19A and go for 19. Is anything showing? There's not going to be really much about the 19. So okay, we go even broader. TMY. Oh, okay. I'm starting to see something to do with TM5. And click on this paper. And oh, once again, we're seeing T4, T5. T4, T5, FI very, very well characterized in the fly vision field. It'll, it'll be unsurprising that you keep seeing these over and over again. Okay, I'm seeing T4, T5. Okay, so now maybe I'll do control. Do this fast track. So, okay, I'm seeing the lobular plate houses things like TMY, this, okay. And just like exploring, okay, I'm seeing 14, 15, 16, 20, not 19. Maybe 19 was something new in the optic lobe that they discovered. And then you can read what the other TMYs do and research things. So that's like one rabbit hole you can find yourself into. And another thing you can do is, again, if you go to the connectivity tab, and if you notice one thing, I'm going from one place to the other, and this is a lot what your research would look like, just comparing, contrasting things. T2 was a pretty big input. So I'm going to search T2 Drosophila. Oh, okay. Seems like there's something on T2. And if I look, okay, T2 are on-off detectors, small visual stimuli. Okay, found something about T2. So now don't know what TMY19A is doing. Seems like it's newly discovered, but I know something about the inputs. I know something about other TMYs, and now I can try to predict what this neuron might be doing and write that in my report. So that is an example of a neuron we don't know anything about. Now, this is another neuron that I will demo, which is one of the DMs. So DMs are one of those neurons in the Fly visual field that we don't know much about, again, because we couldn't really access those neurons to do electrophysiology or these experimental recordings from. But it is a neuron that has intrigued the Fly scientists for many years. And because of the connectome, people have actually started looking at these DM3s, and because of the way they look, the 3A, 3B, 3C, they have proposed predictions for what its function might be. And I think this is one of those neurons which is a beautiful case of how it looks. It's a very, very clear way that they are placed in the visual field. And I will show you as I go along.
Hey,
So
Arushi.
yeah,
Sorry to
yeah.
interrupt. So we have a question that I picked a neuron that projects to the central brain. Those
Yeah.
neurons are, or I guess those regions are not included in the optic lobe. Would it be okay to still work with it? I'm assuming they're referring to the looming neurons, right? Like it projects to the other areas. Like what do you do about it?
Yeah, that's a very, very good question. There is a class of neurons in this optic lobe. Let me go back. So this is the starter guide. If you go here, I explained it here. This connectome paper categorized these visual neurons into 4 types: intrinsic neurons, connecting neurons, projection neurons, centrifugal neurons. It's very likely you will probably stumble on something called the visual projection neurons, which literally take their inputs from the visual optic lobes and project them to the central brain. So their outputs will be in the central brain. Or in the case of centrifugal neurons, you will get inputs from the central brain that go back to the optic lobe. So yes, there will be cases of that. Now, so if you're interested in, let's say, in LPLC2, which is a visual projection neuron, if you actually look at over here, I'll show you one way to know how something is a visual projection neuron. LPLC2. Oh, I think I have skipped it somewhere. Anyways, I won't waste time doing that. But at the top, you have these headings called optic neurointrinsic neurons and like visual projection neurons. So when you probably search LPLC2 in the sidebar, it will show visual projection neurons. So these are neurons that project from the eye to the central brain. Anyways, now your question is, I really like that neuron. Can I still study it? Yes, you can absolutely study it because its inputs will be in the eye, its outputs will be in the central brain. And while I'm not really sure with the accuracy, and personally in my own research, when I look at the outputs of visual projection neurons, they don't tend to be too accurate because this is an optical connectome. So when they were tracing the neurons and actually, categorizing these neuron types, it gets cut off at the point between the eye and the brain. So something like here, it gets cut off. So the downstream, the brain outputs might not be the most accurate, but you can still talk about those, look at the literature if it's making sense, and the inputs will be correct. They will, because they are well categorized and looked into. So it's really up to you. But if you find that it's a bit, jumbled and confusing, you're more than welcome to choose another neuron. There are visual projection neurons that are really well studied and documented. So maybe you will not find this to be an issue because their downstream targets in the connectome will be accurate. But again, I can't really tell you for each one. It's something you just play around and you figure out with. I'm going to assume that's a go. Yeah.
That's great. Thank you.
Okay, cool. Um, yeah, so as I was saying, the DMs, right? So let's look at DM3A. So DM3A looks unexciting compared to the other ones I showed. It's just a thin line in the medulla. And again, the layers and all I won't go over. You guys probably know by now how this works. And then I will go to neuPrint and I will also type in DM3A. And like before I talk about these neurons, you're probably realizing, well, she seems to know like exactly what these neurons are, what they do. And sure, you might think like, but I don't know how I would know that DM3 is something that was understudied. Now everyone wants to know what it does or something, but it's okay. You'll probably pick a DM3 in neuron, and then through your journey you'll come across a paper, then you'll see, oh, like DM3 is considered with DM3A, B, E, C. Like they're all considered together. Okay, maybe I should look at this trio together. And that might be relevant and something. So don't worry, your research journey will inform you of what to do next. So let's go to DM3A. So inputs, outputs localized in the medulla. Let's go to connectivity. Top inputs are DM1, MI4. Top outputs are DM3C, DM3A, DM3B. Okay, so itself and two of its other subtypes. So now I go into the literature. And you look at things. So I actually spelled this wrong. So there's this. But for instance, this is one paper that's actually using the neuronal wiring diagram, which is the connectome, to predict its visual function. And if you look at this paper, you'll be like, okay, predict that 3 DM3s and 3 TMYs. Oh, okay, so there's TMY also in this paper. That might be of relevance. Okay, I see a question. What other thing do you think we can include in the morphology description? Are free to use words like this neuron looks like an octopus with 8 projection, appears like long strands with overshoots at its terminal. That is an interesting question because when you say to someone this neuron looks like an octopus with this, there are so many neurons that look that way, right? So the best way to categorize this morphology is through the ways that I've demonstrated, which is through the neuropils the layers of the neuropils it's intersecting, and the distribution of its pre and post synapse. And yes, you can also say you can say at the beginning, at first when I saw this, it looked like this, but it's important to actually have the terms that I've taught you in this because that is actually the ways connectomic scientists navigate this connectome. So yes, at first you could be like, when I first saw this, it looked kind of crazy to me. It looked like this, but then later on, you actually use this terminology used to explain it. Do you have something to add, Sapo?
Yeah, no, I think that's a great answer. And the idea is we will also want to see your learning journey. So you're free to describe that. But as Harushi mentioned, it's nice to show that you know how neuroscientists actually communicate about describing these structures.
Mm-hmm.
That is very important as well.
Yeah, yeah. So yeah, you can do that. It shows us like this evolution in your learning because at first you're like, oh, it looks kind of wacky. This neuron is kind of all over the place. But then, you evolve into describing, in the proper terms, like its cell body is localized there, it's dendritic arbors, or it's the places where it receives inputs and outputs are localized here and here. Like if you use those terms, it shows us that you have you've learned how to describe things the way connectomic scientists do and how scientists navigate the connectome. So yeah, so this is about DM3s, and so you see this is a connectome paper that is going into analyzing what DM3s does using connectomic analysis. So this is something we don't have electrophysiology evidence for, but still you can, go through this paper and see what this author has done, maybe do something similar and see how your results match up. Look at the 3 DM3s. Do they align this way? What do they look like? And that sort of stuff. So that's another way you can play with neurons. So yeah, basically the neurons that I've picked for the purpose of this exercise, I hope give a varied sort of view of what sort of research journey you might be in. And if it looks hard, I suggest not to give up because you might find something really interesting. And these things are quite, seemingly complex, but when you dig deep, you find that, things might start making sense. And maybe you have a hypothesis that you've created by seeing how this neuron is localized in this larger context. So yeah, I hope that was helpful. If you have any questions, please let me know. But yeah, that's it for me.
Great, thank you.
So just a reminder, if you guys watched the previous recording, you probably know this by now. But again, for those who don't, We have a competition mailing list, so please sign up for it. It's here at tinyurl.com/flymail2025 or scan the QR code, or you can, and also please join our Clematis Discord. A lot of you guys ask really good questions, and when we receive them, we give very detailed answers on the Connectome channel of the Discord. So if you're interested, you can please look at it, and it will probably help you with your own learning. And yeah, thank you so much for attending this workshop. We hope it helped you. We tried to take your feedback from the last one and incorporated new things as we saw that might help. Of course, there are probably other things that we could improve on and to make your learning journey more informative and help you explore this connectomic data. So please, if you have the time, please complete the survey. It will really help us and see what we should do for future workshops and all. Yeah, thank you so much.
Do any of you have any questions? We have a few more minutes, and I'm happy to stay. So someone asked, from my understanding of this workshop, is it better to pick a neuron that's well documented in the literature than a new one? I find it hard to explore a completely new neuron.
Great question. That's a very fair point. It might seem a bit daunting to pick something like TMY19A that we found nothing on. Um, Actually, my personal suggestion for you would be is you should absolutely go and find a neuron that we know nothing about. Why? Because there are already neurons we know something about. And sure, if you want to, you can do that. But I think it's really exciting and it's like real kind of research. If you go find a neuron, and there are many that we know nothing about, and actually like a puzzle solver, try to put this in the bigger context. And even if, let's say, your hypothesis, you feel like, I think like this is not making sense, it's fine. If you try and show us, like, hey, I saw this was the top input and output, and collecting all this evidence together, I think this is what this neuron does, like a detective. That's really impressive. And I bet you would learn so much doing that. So that's my personal suggestion that you should absolutely go for it. In fact, I think you'd find it really exciting to do that.
Yeah, but even with, a neuron that's well known, there's a lot of things you can try. Like if something that's very well characterized, maybe go look at some of the theories behind some of the visual features and the visual behaviors. There's so many computational theories out there that shows how these neurons work as itself and work together with other neurons to detect motion or to give rise to more complex behaviors like courtship and so on. And I recommend like either way works. To be honest, whichever neurons you like or whichever behaviors you feel like studying, whichever circuits you feel like studying, like anything works. So it's really up to you. So meanwhile, you are more than welcome to fill the survey or ask any pending questions, but I wanted to show you something when we have a few more minutes. So I basically want to show you this one, right? In the learning guide, we have, of course, like you understand what the neuron is about. You sort of understand the brief history of a synapse level connectome and how it's being reconstructed. And then please watch these quick videos, like 2 minute to 5 minute videos. They're amazing. They really allow you to appreciate what goes into building this connectome. And of course, we have sort of discussed different aspects of the Fly visual system in the optic lobe, Neuroglancer, neuPrint. But I want to take like a few more minutes to explain this last part in more detail. So as you see, the idea is to allow you to develop some intuition of a neuroscientist and a connectomics researcher. And like Ayushi and I mentioned, when you see some visual scene, there are different visual features. And when the brain processes such features, sometimes or maybe many times it would induce a behavioral response, right? And your goal as a neuroscientist is to study one specific neuron types, and then use Neuroglancer using neuPrint to in the end explain how this neuron contributes within the context of larger circuit to process some information and producing behavioral response. Then we have some resources. And this review is, I mean, there are many reviews, but this is one of the reviews that I think explains quite relatively simply, like what is a visual feature, what is visual behavior, and what are the circuits that's well known out there. So let's go one by one, right? So the first paragraph explains the three learning objectives: visual features, behaviors, and the circuit. So let's go and actually read them. So here you can see, right, animals have eyes. We distill some visual features from the scene so that we can execute an appropriate action, is exactly what we have been saying. Our visual scene contains some features, as you can see, and as this diagram, you see these are visual features. And these visual features leads to some behaviors. For instance, phototaxis is how an animal or an organism reacts to light. Like, does it approach light or does it move away from light? Whatever. So each of these features are explained really, really well in the review, right? And then the goal is try from that review, try and learn some interesting features like optic flow or some behaviors like phototaxis, arousal, courtship, navigation, and so on. But of course, some of this is meant to be for a neuroscientist, so maybe sometimes you'll have to like search a bit more as to what some of these means. But I think it's reasonable for you to go do a bit more research and find out, okay, what does optomotor response really mean if the review itself doesn't explain for like a public audience, right? And here we have like developed like a small exercise for you to go look at the section 5.3. So the goal from 5.3. So the idea is you answer the questions that are outlined in this. This is the main part of the competition, right? Like your goal as a neuroscientist to investigate and answer these three questions. So as an exercise, go to the second paragraph of the local motion detection section. So you go to local motion detection. So it's this section, okay? So you will read what is a local motion detection, why is it useful, and you see it will mention some computational framework and so on. But the second paragraph actually sort of explains some of the neurons that are involved in local motion detection. And you can see a bunch of neurons. T4, T5, like Arshia was mentioned, is such a well-known cell in the lobula. And then there's L1, L2, there's L3, MI1, TM3. So there's a bunch of neurons. But if you actually try, and let's say you're like, I fancy to study L1, and I want to know how this play a role in motor detection. The first thing you should do is, okay, where's L1 in the optic lobe? Where does the L1 output its neuron based on the review? And does it match with what you see in the Neuroglancer and the neuPrint and so on? So this is some sort of exercise that Archie has been showing you. And in the resource guide as well, we sort of try to write it down in text. Hopefully one of the ways of communicating works for you. And as always, if you have any questions, hopefully that helps. Okay.
Can I say something as well? I just thought of something that might be helpful. So I'll just share my screen. So this is a paper that is comparing the on and off edge pathway, which is the pathway involved for T4, which is the on edge pathway, and T5 off edge. It's okay if you don't know what this means, but for instance, let's say you find a neuron and that is, I don't know, TM2, for instance, right? You could go into these papers and look at their figures. And because they've already curated, and this paper did the connectomic analysis, they curated all these neuron networks. You can see, okay, I see TM2 is like down here and it's inputting to T5, and this is like the network is around. Or if you find a neuron through your neuPrint, which is somehow connected to like MI1, but it's like not on this diagram, you can like put it into like context. I see, okay, so in this figure, this is what we know, and this is how it's connected. These are the mechanisms these guys have proposed for this neuron. And my neuron, this new connection is somewhere around here, so maybe it does this. So it's really helpful to use these figures you will find in papers, and you can sort of like add a sticky note of your neuron and where you think it places into this and how it works. Just a small thing I thought that might be helpful as well.
Yeah, thank you. So what you're saying is there are these figures. That's one example, but many papers will have these kind of proposed circuits. And if a neuron is not in that circuit but connects to some of those well-known ones like T4, T5, L1, MI,
MI1 or something like that, yeah.
yeah, or the TM's neurons, then you can sort of try and figure out where in the circuit does it play a role and identify like does it process some visual features, what exactly does it do, how does it do it, how does it actually provide a mechanism that allow the fly to produce some behavioral response. How exactly does it do it? Like, don't just say like it helps detect motion, but try to go into more detail. As far as you try to uncover new insights, that's the better, right? You sort of learn. Okay. How do we systematically report how we identify and choose the neuron? So this is up to you. You have 1,000 words. You and of course we want you to show your learning journey. It's really up to you, right? You can if you feel that that learning journey was really important on how you choose the neuron, that is fine too. Okay, for choosing the neuron, It's really up to you how you choose the neuron and how you want to report it. That's okay. And you can show it in either the creative piece or you can show it in writing. In terms of how you report, you mean like the morphology, or you just mean like how you actually choose one of the neurons? There's another way in that, let's say you're like, I like motion detection, and I found it seems like there's a gap in this paper. And you're like, and I've noticed this neuron seems to play a lot of role in the motion detection circuit. No, you don't really need to do systematic review, no. The point is we care more about you actually using the connectomics tools, so you don't really need to use the Prisma framework. Arshi, did you want to explain something?
Oh no, I just brought this resource up to show if you're not sure how to like construct and lay down your ideas, we have a research report template here with our application guideline to give you, an idea. Okay, maybe this is something I should include. You can always include more than this and things like that. But yeah, this is a brief guide on how you could write your report.
Okay, we have another question. This is a medical student who wants to do a residency, and they're asking that does this project align with their learning goals of becoming a resident, like an aspiring neurologist? And so does this align with the fact that someone who wants to do a clinical research? So I can answer. I think it does. So I use a lot of connectomics in psychiatry and, to study mental health disorders. And then there are other people who use sort of computational neuroscience to study different brain disorders. So I think the fact that you know how to analyze a large scale brain data set is in itself an important part of neuroscience. And remember, or like most of the clinical questions, they are often based on animal models like Drosophila, which is the fly, or the rats. And most of the times, like you sort of have to manipulate some neurons and do some experiments. So that's the experimental side of things. But now with the release of this large scale data set, both experimentalists and the computational neuroscientists often have to now rely on this large scale data set in order to find new questions about the brain and so on. So I think, yes, it aligns really well with anyone who's interested in clinical research. I hope that makes sense. Great. Anyone has more questions? I'll give it one more minute. But I think I'll ask Arushi one question. So when you started learning about this connectome, right? Like it's not like everyone was born with the ability to analyze the connectomics dataset. So I want you to take us through a little bit of how you actually got started and how you found the courage to learn all of this. Like I think it's amazing that you can actually learn from not knowing anything to suddenly now being pretty much gain a huge sense of familiarity with the connectomic data. So can you like outline a journey from where you start and
Yeah, sure. No, it's fair because I my academic background is not even in neuroscience. It's actually in biochemistry. So as someone who didn't really even know about how neuron really works, it was quite a jump. And um, so I remember first when I joined this field, my supervisor would tell me this field is new and you have to get lost in the data. And I was like, what does that mean? I don't even understand. These neurons all look the same to me. They look like a bunch of cauliflowers, trees, and I safe to say literally every time, what is this? And it was confusing because it felt like staring at paint dry sometimes. But when you analyze these data, when you read about discoveries, I personally found like how Drosophila does courtship very interesting. It's just this fascinating communication and the behaviors they display and like neurons playing this role. Then I started again like this behavior. I got so interested, like these tiny things that do like action potentials can somehow guide something so complex. And then it got to a point when I'd go to Neuroglancer and look at it, I'd be like, oh wow, it's so interesting. It's in this part, it's projecting to the brain, or it's more like it's a simpler thing. It's more up in the medulla. It's integrating inputs at a more like simpler level, just getting information. Or when I'd look at connections, it's because I do these analysis using Python and like coding, I would see, oh, like this neuron is connected to this, and it's doing this sort of connection. Oh, that's really interesting because it has some reciprocal thing going on and stuff. And like the more you keep looking at it, the more it sort of becomes a bit magical how uh, every neuron has its own character to it. It has its own flavor, it has its own thing it likes to do, and it gets really, really interesting because at first it's kind of hard to appreciate because like I think someone said this looks like an octopus and it's very fair. That's what it looks like to me as well. Then you start to see like, oh wow, this is cool. And then you start to go like the people who made this, the people who actually like looked at this and said, this neuron is different from the other. And you just, go into this rabbit hole and you start thinking this is incredible. This is an incredible sort of science. So yeah, that was my journey. And yeah, now I think it's very cool. And yeah, it's very interesting.
Great. We have another question that I was messaged. So, does a connectome for human brain exist yet? Before answering that question, I want to expand more a bit on the implication of studying the fly connectome for a medical researcher, right? So, I think there's a few things. So, first is I think the transferable skills. Remember, as a neuroscientist, you will often use a large scale brain data set. And I think that in itself is quite transferable to any neuroscientist or neurologist and so on. And I think it's very cool. If you go look at physician scientists at research institutions, many of them are actually computational neuroscientists or they're molecular neurophysiologists or whatever, right? They use fruit flies, they use mouse, and they study cool things using both experiment and computation. So that's the first. And the second is remember fly and rats, these animals try to survive just like us. And if we understand how the fly have neuromechanism that ensures survival, like courtship depends on the visual information that it receives or like detecting objects and so on. So by studying how the brain ensures the animal survival, you can always study what happens if that goes wrong. It's very similar, right? So that same logic applies as well. And the third is that these complex behaviors, they're conserved across species. So that's the medicine part. But then there's another question on, is there a human connectome? So I want to share this slide from our workshop one. Can you see this part? What is a human connectome? You can see my slide, right?
Yeah, I can.
Perfect. So keep in mind that the Fly optic lobe connectome is one of the most comprehensive wiring diagram in the sense that we have the detailed map for every single neuron, I want to emphasize it. And we also know exactly the synapses, like the connections, those that dendritic arbors that you see, right? And like here, as you can see, we can see like exactly specific neurons, where is it connecting, and so on. But the human connectome, so yes, we do have many human connectomes, but we do not have a synapse level human connectome, at least for the entire brain. Maybe there's like 1% of the brain or something, but it's not,
Yeah.
it's currently, we haven't gotten that far yet. Only recently we have like the fly, like the full brain of the fly, and we have some part of the mouse brain, but that's about it. But to answer your question, but what's a human connectome then? So a human connectome is created when we, like as human beings, lie on the MRI scanner. And then in the MRI scanner, you can have different settings. So in one of the setting, we call it the diffusion tensor imaging. So here you can measure white matter tracks, which are basically bundles of axons that connect different regions of the brain. But keep in mind, these axons have probably like thousands or millions of neurons in it. So it's really a mesoscale sort of connectome. It's not synapse to synapse level. So when you image these white matter tracks, we call this a human structural connectome. But unlike the fly connectome, we currently do not have the synapse level resolution connectome yet. We are still making progress, but at the moment, one of the most comprehensive wiring diagram at the synaptic level is the fly brain. So we have like the whole fly brain, and for the competition, we're using optic lobe, partly because fly dedicate half of this brain to vision, and vision is just, it links to pretty much a lot of behaviors out there. I hope that answers the question about the human connectome. Okay, it seems like we don't have any more questions. So thank you so much, guys, for coming to our workshop. Bye bye, everyone.
Clematis Research Empowerment Hub. Empowering research for everyone.